Hi there, I'm Kylie, and I'm here with Jasmine and Aveen. Our presentation will be on the investigation of the physiological effects of metformin. We think that there will be a wide variety of genes expressed when metformin is administered. The objective is to understand the magnitude of the effects that metformin has on organisms, specifically regarding gene expression. Metformin is a blood glucose lowering drug. In our project, we are going to use GWAS and GEO to further understand the effects of metformin on organisms.
In the beginning of the year, for my honors thesis project, I performed an experiment that assessed the effects of metformin on birds, specifically mourning doves, and we checked their blood glucose levels. After the administration of metformin, we initially assumed that their glucose levels would decrease because that's what metformin was supposed to do, but actually their blood glucose levels increased significantly compared to the control after 150 milligrams of metformin. And so the main purpose of this study is to assess why this happened in the birds.
So for our methodology, we used two different computational technologies. The first one being GWAS, which was utilized through a control study that compared and analyzed the SNPs of the entire genome of a large sample size, in this case avian birds, with a sample group with a specific disease, in this case diabetes, against the secondary avian group without diabetes. Ultimately, this was just in order to better understand the effects of metformin on the animal body. After doing this research, we were able to discover 4 matching SNPs, which were then scientifically proven to correlate with diabetes through other proven studies. So to sum it all up, pretty much we were just looking at how diabetes correlates with different immunoresponse treatments, such as producing beta cells in the pancreas. And with this, we were able to correlate it with the GO2R, which was essentially used to find a pattern of gene expression after the administration of metformin. Relevant studies were then analyzed and extracted and ultimately showed a significant difference. This will potentially allow us to find a pattern to be able to better understand the effects and response of metformin.
So for the first half of our results, we looked at human macrophages, and with the administration of metformin, there was no significant difference shown. The table to the left shows the gene expression that is related to asthma, and it has no significant response when metformin is administered, just like all the other data sets.
So as we established before, it's well known that birds maintain high blood glucose levels, but a different data set was retrieved for mice. And in this mice data set, specifically that showed liver expression and gene expression, we can identify that a major category of genes that were expressed were genes that were related to immune response. And one pattern that we're able to identify was pretty much the absence of a pattern, which means that after metformin was administered, there was a wide variety of data sets that showed a significant difference. But the only thing we were able to identify was that certain genes are turned on or turned off with the administration of metformin. For example, lipocalin, which is a gene that is involved in inflammation and detoxification and immune response, is significantly active when metformin is administered. The same thing with the hepatitis A cellular receptor. But other genes such as cytochrome P450 and diacylglycerol kinase, which are also involved in immune response in some way, specifically detailed on the poster, the expression was significantly less or inactive with the administration of metformin. And so one of the major limitations of GWAS and specifically GEO first is that it's limiting ability to provide additional methods of analyzing next-gen sequence data. And the limitation of GWAS was its inability to look into low blood glucose level SNPs due to the SNP focus being on the correlation with diabetes and the chosen specific genes. So all in all, we can see that after the administration of metformin, specifically in mice, that they either turn on a gene or they turn off a gene. And these genes seem to be involved in immune response. But then again, there are so many other genes that are activated and so many other pathways as to why the birds in my thesis project showed increased blood glucose instead of decreased. That being said, we do know that metformin is to be considered a dirty drug because of its vast effects of physiology on organisms.